Welcome back, dear students. In today's video, I am going to discuss a topic from the chapter Human Health and Diseases. And in this chapter, one of the most important topics is mechanism of humoral immunity and cell-mediated immunity. And to understand what exactly humoral immunity and cell-mediated immunity, first we have to know what are the most important type of cells involved in the immune reactions and immune responses. And when you look into the different types of WBC which perform different types of functions, out of all the types of WBC, the most important cells associated with immune system are lymphocytes. And if you remember, in blood composition we have studied totally 5 types of WBC are present. Out of the 5 types of WBC, the second most abundant type of WBC are the lymphocytes, which are one of the most important cells of our immune system. And these lymphocytes are categorized into 2 types, namely B lymphocytes and T lymphocytes. And we have to know in detail what exactly B stands for and T stands for. So here students should understand all the lymphocytes, whether it is B lymphocyte or T lymphocyte, ultimately lymphocytes are produced from the bone marrow, because bone marrow is the hemopoietic organ. But some of the lymphocytes, after their production in the bone marrow, they differentiate into functional cells in the bone marrow itself, and that lymphocytes are called B lymphocytes. So that's why B stands for bone marrow. And some other lymphocytes, anyways they are produced in the bone marrow, but they are produced as immature cells, non-functional cells. So some of the lymphocytes, after their production in the bone marrow, they migrate to thymus. Under the influence of the hormone thymosin, they transform into functional cells. So with this it is very clear, both the types of lymphocytes are produced in bone marrow itself, but based on their differentiation into functional cells. If the lymphocytes are differentiated into functional cells in the bone marrow itself, they are called B lymphocytes. If they are differentiated into functional cells in the thymus, they are called T lymphocytes. So as here I was telling you, basically immunity can be of 2 types, that is humoral immunity and cell mediated immunity. So broadly speaking, humoral immunity is offered by which type of lymphocytes? B lymphocytes. So B lymphocytes offer us humoral immunity, and ultimately which type of cells offer us cell mediated immunity? That is T lymphocytes offer us cell mediated immunity. So with this introduction, I want students should understand, whenever a foreign antigen enters our body, our immune system elicits 2 types of immune responses. They are broadly classified into humoral immunity and cell-mediated immunity. Broadly speaking, humoral immunity is the function of B lymphocytes. Cell-mediated immunity is the function of T lymphocytes. Now, let us look into the details of mechanism of B lymphocyte action. And if you remember, I already told you, B lymphocytes offer us which type of immunity? I already told you it is humoral immunity. And if you ask me, what is this humoral immunity? My simple answer, the immunity offered by the different body fluids. Actually humor stands for fluid. The most important body fluid what we have, that is our blood and blood plasma. They contain different types of antibodies, and these antibodies fight against the foreign microorganisms or the antigens which are entering our body. So whenever we get an immune response or whenever we get resistance against a disease by the antibodies, ultimately we have to understand that is nothing but humoral immunity. But humoral immunity, simply students should remember, the immunity offered by the circulating antibodies present in the blood is called humoral immunity. and from where these circulating antibodies are produced. I already told you who will produce antibodies. Broadly speaking, antibodies are produced by the B lymphocytes, and how and when these B lymphocytes produce the antibodies. Let us look into the mechanism now. Let me draw a simple B lymphocyte here. Usually the B lymphocytes on their surface, they are provided with a marker, and this marker acts as receptor for the antigen, and this marker is called BCR. Name itself indicates it is the marker present on the surface of B cell. That's why it is called B cell receptor. It acts as receptor for what? It acts as receptor for the foreign disease causing antigen. So whenever a foreign disease causing antigen enters our blood circulation, and this marker recognizes the antigen, and antigen moves towards the marker, and antigen finally binds with the surface marker. Whenever antigen binds with the surface marker, now the B cell, that is B lymphocyte, undergoes differentiation, and this differentiation requires a type of cytokines, and this type of cytokines are called interleukins, ILNs we say. So the type of cytokines responsible for the differentiation of B lymphocytes are called interleukins. If you remember, interleukins are produced by the type of T lymphocytes, that is T helper cells. T helper cells will produce low molecular weight proteins called cytokines, which help in the differentiation of B lymphocytes into two different cell populations. What are those cell populations? They are memory cells and plasma cells. So students should understand this mechanism. This is a B lymphocyte. On the B lymphocyte, what is present? On its surface there is a marker. Marker means what? It is the receptor for an antigen. The receptor present on the B cell surface is called B cell receptor, and with the help of this receptor, B cell directly can recognize an antigen and it binds with the antigen. Whenever antigen binds to the BCR in presence of type of cytokines called interleukins, B lymphocyte differentiates to form two different cell populations, which are the memory cells and plasma cells. And these plasma cells otherwise called effector cells, because now the plasma cells are only going to show the response. What is that response? Production of antibodies. So with this it is very clear. Antibodies are actually produced by whom? Antibodies are produced by plasma cells. But from where the plasma cells are formed? Plasma cells are formed as a result of differentiation of B lymphocyte. So B lymphocyte differentiation occurs always in presence of what? Interleukins. Who will produce interleukins? T helper cells. Now plasma cells are otherwise called effector cells. They produce the antibody, and now this antibody will go and bind with the antigen, and it neutralizes the action of antigen or destroys the antigen, and it makes our body free of infection. And then what about these memory cells? Memory cells do not show any function in the first exposure, but they generate immunological memory against this antigen. Let me consider this antigen as A. Now this memory cells will generate memory against what? Antigen A. So if the same antigen called A enters your body or affects your body for the second time, all these process is not required. Already memory cells are there. Immediately they get transformed into effector cells. They produce the antibodies, and antibodies tackle the antigen. So this is how B lymphocytes ultimately, by producing antibodies. Antibody is nothing but an antigen binding protein, and antibodies are always specific for an antigen, and these antibodies freely circulate in the blood and they offer as a type of immunity called humoral immunity. Now let us look into the mechanism behind the cell mediated immunity. So cell mediated immunity is simply called CMI, and broadly speaking cell mediated immunity is offered by T lymphocytes. Already I told you the lymphocytes which transform or differentiate into functional cells in the thymus are called T lymphocytes, and which are known to offer cell mediated immunity. And the first and most important point according to syllabus students should remember, T lymphocytes are the cells responsible for graft rejection or immune rejection. So it is very simple. The type of immunity responsible for rejection of the organs which are transplanted, then our answer should be cell mediated immunity. So cell mediated immunity is offered by what? T lymphocytes. So it is very clear, T lymphocytes are the cells responsible for immune rejection or graft rejection. T lymphocytes offer us which type of immunity? Cell mediated immunity. Now when we go into the details, T lymphocytes are further classified into two main types, that is Th cells and Tc cells. Th stands for T helper cells, Tc cells stands for cytotoxic T cells, and this is based on their function, and it is also based on the surface marker present on the cell. And T helper cell is provided with a surface marker on it, and this surface marker is called CD4 marker. That's why T helper cells are also called CD4 cells. If you remember, when I told you the action of B lymphocyte on the surface of B cell, there is a receptor for antigen. I called it as what? BCR. And similarly, on the surface of T lymphocyte also a receptor for antigen is present. I call it what? TCR. That T cell receptor can be of two types. It can be CD4 and CD8. On the surface of T helper cell, which type of marker or receptor is present? CD4 marker is present. That's why T helper cells are called CD4 cells. Whereas on the surface of TC cell, which type of surface marker is present? CD8 marker is present. So T helper cells are otherwise called CD4 cells. TC cells are otherwise called CD8 cells. When we look into the name, T helper, these are the cells which help all other cells of the immune system. How can they do that? T helper cells are responsible for the low molecular weight proteins which are called interleukins. Interleukins act as signaling molecules, and they also help in differentiation of non-functional immune cells into functional immune cells. So with this it is very clear, if the T helper cells are not there, most of the immune cells cannot transform into functional cells, and the level of immune response keeps on decreasing day by day. If you remember, this is what happens in HIV AIDS, because in HIV AIDS, day by day, month after month, which type of cell count decreases? T helper cell count decreases, and all other cells also fails to perform their function. So slowly and progressively person develops immunodeficiency. So with that it is very clear, students should remember, T helper, they help all other cells of the immune system. How can they do that? By performing or by producing a special type of chemicals called interleukins. And TC, cytotoxic T cells, and these are the cells which offer us highest level of immune response. Highest level of immune response is called cytolysis. Name itself indicates. Cytos means cell, lysis means destruction. Just imagine, in our body, in any tissue, if very few cells are subjected to viral infection and these viral infected cells are lysed or destroyed in a process called cytolysis, and it is ultimately done by whom? TC cells. That's why they are called cytotoxic or cytolytic T lymphocytes. On their surface, which type of receptor is present? CD8 receptor is present. That's why they are called CD8 cells, and T helper cells are called CD4 cells. And now let us understand how this T helper cell can produce interleukins. So I have drawn a very simple diagram of a T helper cell. On its surface, what is present? Simply I can call it TCR, that is T cell receptor. But which type of TCR it is? CD4 marker. That's why T helper cell is otherwise called CD4 cell. And here students should remember, in the mechanism of B lymphocyte action, what I said? B lymphocyte, with the help of its receptor called BCR, directly it can recognize an antigen, but T lymphocytes are not having that capacity. Always students should remember, T lymphocytes cannot recognize an independent antigen. If they are unable to recognize an independent antigen, then what do they recognize? If you ask me this question, my answer, they always recognize antigen plus a special protein complex called MHC. So T lymphocytes always recognize antigen and a special protein complex present in our blood, which is called MHC. MHC stands for major histocompatibility complex, and this major histocompatibility complex is of two classes. We call it MHC class 1 and MHC class 2. So let me repeat, B lymphocytes can directly recognize the antigen, but T lymphocytes cannot recognize the antigen directly. Then how do they recognize the antigen? They recognize a complex. What is that complex? Antigen plus MHC. MHC stands for what? Major histocompatibility complex. It is a special protein complex present in the blood. And MHC of how many classes is present? Two classes is present, that is MHC class 1 and MHC class 2. Now let us look into the details of action of T helper cells. If you remember, helper, they help all other cells of the immune system, I said. So how can they help all other cells of the immune system? By the production of low molecular weight proteins, we call them interleukins. Interleukins are essential for the differentiation of non-functional immune cells into functional immune cells. Now let us understand how and when a T helper cell produces interleukin. See, here I have given a clear representation. So this is T helper cell. On the surface of T helper cell, which type of marker is present? CD4 marker is present. I already told you, CD4 marker cannot recognize the independent antigen. It always recognizes what I said. It recognizes antigen plus MHC complex. And you can see here, this is the antigen, and where antigen is present now? Antigen is present attached to MHC. MHC of which class? MHC class 2. And you can see in the diagram, MHC class 2 is present on the surface of a special cell called APC. APC stands for antigen presenting cell. So here students should understand, in our immune system, we are gifted with a special type of cells called antigen presenting cell. Whenever foreign antigen enters our blood circulation, this APC will engulf that antigen and process the antigen, and the processed antigen is displayed on its surface, because on its surface what is present? MHC class 2 is present. So students should understand, on the surface of antigen presenting cell. Name itself indicates. This is the cell which presents the processed antigen to whom? T helper cell. And in the diagram you can see now the antigen which is processed by APC. It is attached to what? MHC class 2 present on the surface of what now? APC. So if you ask me, CD4 markers can recognize what? CD4 markers present on the T helper cell always recognize antigen plus MHC class 2 present on the surface of APC. When CD4 marker recognizes, CD4 marker binds with antigen present on the surface of APC. Once CD4 marker binds to the antigen, T helper cell differentiates into effector cell population. Effector means nothing but functional. And what is the function? I already told you. They produce the low molecular weight proteins. These low molecular weight proteins are called interleukins. These interleukins act as signaling molecules, and interleukins also help in differentiation of non-functional B lymphocytes into functional B lymphocytes, and also they help in the differentiation of non-functional TC cells into functional TC cells, which can perform the function of cytolysis. So this is how T helper cells play an important role in the functions of various cells of our immune system by producing chemicals called interleukins. Now let us look into the action of TC cells. I already told you, on the surface of TC cell which type of marker is present? CD8 marker is present. So I can call TC cells as CD8 cells. If you ask me, sir, what is the function of TC cell? My direct answer is TC cell offers us highest level of immune response. And what is highest level of immune response? Highest level of the immune response is called cytolysis. Lysis is destruction of the cells. Which type of cells? Just imagine in your body tissue, one cell is subjected to cancer or virus infected a cell, and that virus is multiplying inside one of the cells of your tissue. And just imagine, tissue is the group of cells where thousands of cells are there. Just imagine 10 cells are already infected with virus. These 10 viral infected cells are lysed or destroyed. Why? To make all remaining cells of your tissue free of the infection. So this is said to be cytolysis. So students should remember, healthy cells of our body are not subjected to lysis. They are not destroyed. When our immune system is normal, immune system function is normal, which type of cells are subjected to lysis? Infected cells like cancer infected cells, viral infected cells are lysed by TC cells to make other cells of the tissue healthy and free of infection, and that is the ultimate and highest level of immune response, which is called cytolysis, and it is offered by which type of cells? TC cells. And now let us look into the mechanism. So TC cell, on its surface, which type of marker is present? CD8 marker is present. And once again the question, CD8 marker recognizes what? My answer is clear here. CD8 marker recognizes, this is the antigen. You can see in the diagram where the antigen is attached. Antigen is attached to MHC class 1. MHC class 1 is present on the surface of what? ASC. Once again students should know what is ASC. ASC stands for altered cell. And if you ask me, what is altered cell? Name itself indicates any cell of our body which is altered, changed. What is that alteration? That is nothing but infection. Just imagine, in your body tissue, very few of the cells are subjected to viral infection. Virus is multiplying in those cells, and I call those cells as what? Altered cells. So any infected cell of the body is said to be what? Altered cell. On the surface of altered cell, which class of MHC is present? MHC class 1 is present. And once again you can see the antigen which is entered our body, it was engulfed by the cell and it is processed. Now the processed antigen is displayed on what? Class 1 MHC present on the surface of ASC. So with this, student should remember TC cells recognizes antigen plus class 1 MHC present on the surface of ASC whenever TC cell recognizes this complex, that is antigen plus class 1 MHC complex. TC cell undergoes differentiation and forms effector cell population, and this effector cell population is called CTL, cytolytic T lymphocyte. CTL stands for what? Cytolytic T lymphocyte, and non-functional form is called TC cell. TC cell on differentiation transforms into effector cell, and now this CTL are going to perform what? Cytolysis. Cytolysis of what? Altered cell. So now already in the diagram you can see this cell is, for example, it is already infected with virus. Now this virus is getting multiplied inside these cells. Now this CTL cell will approach the viral infected cell. On approaching viral infected cell, what should happen? Tell me. It should be destroyed or it should be lysed. And who will perform this function? Some enzymes. Whenever CTL approaches the viral infected cell, it produces different enzymes like cytolysins, perforins, etc. So name itself indicates perforins. They cause pores on the surface of altered cell. Then cytolysins through these pores penetrate the cell. Now these cytolysins are the very strong enzymes which lyse the altered cell. When the altered cell is lysed, inside the altered cell what are present? Tell me. Virus was present and virus was getting multiplied. When the altered cell is lysed, destroyed, the virus which is multiplying inside the cell also destroyed. So neighboring cells will be healthy and they will become free of viral infection. Such a magnificent and wonderful mechanism we are gifted with, which is called cytolysis, and it is the function of type of T lymphocytes called TC cells. So TC cells on differentiation, they transform into effector cells called CTL, and this differentiation once again requires what? ILNs, interleukins, which are produced by T helper cells. So with this you can understand, if T helper cell count decreases, amount of interleukins produced also decreases, and even the degree of cytolysis also decreases. So viral infections, cancer infections persist as it is in the body. So that's why T helper cells are very, very important. That's why I am telling you the topic. In HIV/AIDS, whenever T helper cell count decreases, functions of all other immune cells of our body systems also disturbed. So this is how to offer us two important types of immunity. What are the two important types of immunity? I already told you, that is humoral immunity and cell mediated immunity. So broadly speaking, who offers us humoral immunity? B lymphocytes. By what? By producing antibodies. What are antibodies? Antigen binding protein molecules. CMI, cell mediated immunity, is offered by whom? T lymphocytes. What are the two important type of T lymphocytes? T helper cells and TC cells. Let me repeat. T helper cells are called CD4 cells and TC cells are called CD8 cells. What is the function of T helper cells? Production of chemicals called interleukins which help in the differentiation of non-functional immune cells into functional immune cells, and TC cells on differentiation offer us highest level of immune response, that is called cytolysis. Other than this, TC cells and T helper cells, we have other types of T lymphocytes also, but students should remember one more type, that is called T suppressor cells will be there. Name itself indicates. With this explanation I said, if a foreign antigen enters our body, immune system recruits variety of cells and different types of cells will be showing different actions. At the end of all these actions, what happens, tell me? Our body becomes free of infection. Once the body becomes free of infection, do you expect any more immune response to be required? No. So that's why once the body becomes free of infection, immune response should be suppressed, and it is done by another type of T cells which we call them T suppressor cells. So in this video I have explained you what is humoral immunity and what is cell mediated immunity. Broadly speaking, humoral immunity is offered by B lymphocytes by producing antibodies. Cell mediated immunity is offered by T helper cell and TC cells by producing interleukins, and finally by performing cytolysis. Hope you understand and like this video. Thank you.